All right, hey everybody. So I forgot to videotape during class, so I'm just gonna do it quick right now. So we talked about the rest of the notes today, and then just things, it was isomers. We started talking about isomers and kind of reviewed that, and then talked about, we're remembering it has the same chemical formula, just different structure. So there's three types, and that's the first one. That's structural isomers. And so as we can see on here, these are all single bonded carbons, and that's kind of a big thing to look at for these. And we're just taking one of these carbons and putting them in a different place. It could be in a ring structure as well, but structural isomers is changing the structure, nothing really else. When we're looking at geometric or cis trans, now we're off of a double bonded carbon in there, and in this example these X groups would be a possible functional group that you're learning right now, or maybe an oxygen off of there.
Landon Bain to the office, please. Landon Bain to the office.
Okay, and then so anyway, we just take that and it's like we're turning that carbon and we're just flipping. But the flipping of that oxygen on that molecule, again, structure determines function, changes the structure, then changes the function. And that's the biggest thing with that. It still has the same chemical formula, but can have different functions then. And then just so you know, cis trans and geometric are talking about the same thing, just two words for that type of isomer. So you could see it either way, I guess, just know that. Then this last one, our stereoisomers, is like we have these two versions and it's mirror images of each other. It's like if you take your hands and you put one over the other, right? Our thumbs are on the opposite sides. But when we're talking about enantiomers, we take our left hand and we're going to put our hand on a mirror, then our thumbs would be touching and our fingers would be together. So these isomers are really like flipped isomers, but they're mirror images of each other. And then therefore, when we straighten those out, we say that this side has my pinky, this side has my thumb, this side has my thumb, this side has my pinky. Anyway, that goes to why they call them the left-handed and a right-handed isomer. And so if we look at these, the ones you want to remember are left-handed isomers all the time. Those are the biologically active, or also what we would call maybe good isomers, because you've probably already gone through these notes and seen the other things that they could be. But this next slide gives us examples. Really, no, left hand, left version, that's the one that we use, okay? So examples that could be like an amino acid alanine. We use the left for that in proteins, but not the right-handed version at all. In medicines, the L version is active against Parkinson's disease, the right-handed non-functional at all. Structure determines function. But sometimes we can talk about things with tragic results as well. So when we look at this, we have methamphetamines, or meth. Meth is the right-handed version of what we use on the other side, the left-handed version for nasal decongestants that you can buy over the counter. You take that and then you make it a right-handed version, and then you start talking about making meth. Don't do drugs. Meth is bad. Another one is thalidomide, and the left-hand version is used for really great things like cancer treatments, Crohn's disease, also for some things for HIV. And if you take the right-handed version of that, which was actually given to women in the 1950s for they were pregnant women who had morning sickness, so they're given for the effects of nausea, and it helped with the effects of nausea, but then they found it had detrimental effects, and that their children had abnormalities when they were born. So when we're talking about enantiomers, left-handed versions are the ones we use, right-handed versions are either the ones that are non-functional or probably going to cause some type of problems. Let's see. Okay, so just remember there's three different types of isomers. Number one, structural, that is single bonded carbon. Then there's a second one, which is cis trans. or geometric, that's double bonded carbons. And then we have these stereoisomers or enantiomers, which left-handed is good, right-handed is bad. Okay, and then this is what we're getting to for functional groups. So we're gonna go through in the next chapter, chapter 5, and talk about all those biological macromolecules. And when we talk about that, it's really a carbon structure that then adds a functional group to it. Oop, I'm standing in the way. Okay, so once we take something, this is the simplest form that we talked about, those hydrocarbons, we exchange one of those hydrogens for a functional group that actually changes that whole thing to something else. So now we're talking about ethane to ethanol, and now we can start to gas our cars. Or there's also ethanol, and that's going to have the sulfhydryl group attached to it. So we looked at functional groups, and this example, which is in your notes, I told them you like really want to know, probably keep this example in your mind, is for functional groups. A really great example is male and female hormones. I feel in the way, you don't really need to see me, I guess. It's male and female hormones. So both of these are steroids. I mean, they're steroid based. And when we're talking about the female hormone, they have a hydroxyl group off of it. And we can see that both of them have this methyl group here and this hydroxyl group over here. But when we're talking about estradiol to testosterone, we see that testosterone is different because it has an extra methyl group attached, and then instead of a hydroxyl here, it has a carbonyl group. And so we have a very similar structure, but we're just changing those functional groups on the outside. And now we're talking about one hormone that looks like this that causes female characteristics. You change a couple things, and now we're talking about a hormone that is causing male characteristics. And so when you're talking about just changing functional groups, like it sounds like such a nonchalant thing, like, oh, we're just going to change a functional group. That creates a huge difference in whatever we're talking about that's working in living things. Okay, so those were the 7 different structures that you were looking at. Hopefully you can still hear me. And when we're looking at these, hydroxyl is one of them. And the big things off of that is an OH off of there. What do you want to know? It is polar. So if we're talking about polar, that's going to be soluble with water. Names are going to end in OL, and we're looking at alcohols, so like methanol, ethanol, ending in ol, isopropyl alcohol. All those are examples of something that would have a hydroxide group attached to them in order to make those different compounds that we're making. Okay, carbonyl and carboxyl. I'm going to kind of flip between these because these two are the ones that probably people get mixed up the most. And so something that I want you to keep in mind is that when we're looking at this, you can see that this has a hydrogen on there, but there's 2 different types of carbonyl groups. Carbonyl groups can be in the middle of a structure, or they can be at the end. And so that's when we get to these other terms on here that we call aldehydes and ketones. And so I'm gonna, like, my way to help you remember these 2 is, and I made the class say this, you missed a really good time, but aldehydes oxidize, and that's up there, so it rhymes. Aldehydes oxidize, and just keep saying that in your head. And when we're oxidizing, that means it's at the end. So we can see this in this top structure up here. And what I think is just like, okay, so we have this carbonyl group with an oxygen, double bonded oxygen attached to it. And when we're oxidizing, we need air to go through, right? And it has to be on the end of something for like rust to form. But aldehydes oxidize, they're at the end. And then the ketones, they're in the middle, so they're more hidden, so they can't be oxidized and hit with air. And I think of keys with the prongs, right? So then ketones are gonna be in the middle of a carbon structure, and that's where they're gonna stay. So aldehydes oxidize, say that 10 times fast. And then ketones, that's like a key, and it's a little bumps on the key, and that's gonna be in the middle of that carbon structure. Now, when we look at carbonyls, then this one that we're seeing here, that is going to be an aldehyde. That's on the end. That's with a hydrogen. But as you can see, these ketones, they don't have an oxygen attached to them. So some people, they're like, I need the hydrogen on there, or I don't need the hydrogen on there. It doesn't matter. It's really where it's located if it matters. And if you just circled the carbon and double oxygen, that's all that really matters for the carbonyl group, okay? So carbonyl and carboxyl are the ones that get mixed up the most, because we'll look at carboxyl next. And for carboxyl, it's the same thing. We have a double bonded oxygen, but now we're to a hydroxyl group or an OH. My dumb way of remembering this is carboxyl just sounds fancier than carbonyl, and therefore it just has more stuff going on. Okay, and we have this OH group attached to that. Sorry, I just thought of something. So carbonyls, something you might want to add to that is that carbonyls are going to be found in sugar structures. And so like their whole function and purpose, well, I don't want to say whole function, but one of their function and purposes is to, they say like it's for aromatics or to make things smell nice, and then also to taste sweet. And that's what you think about with sugars. So whenever we look at sugars, which is a carbohydrate, which we're going to look at, we're going to see carbonyl groups on those for sure. And then when we see carboxyl groups on here, and it says this in these examples here, but we have the word acids at the end. Sorry, I don't know what's happening. Anything that has the word acids at the end, that's going to be in a carboxyl group. So I'm probably not going to give you a name of something for a carbonyl group. I don't know. I'd probably just say something like think about sugars or think about smelling really nice. Those are kind of things for carbonyl. Carboxyl, if it has the word acid, then we're looking at a carboxyl group. And actually, when we start looking at amino acids, which are building blocks of proteins, we're going to see the next functional group, I believe, which is amine, or the amino group. And then that's going to be attached to a carboxyl group. And then we get amine group, acids, carboxyl group, put those together, amino acids. Okay, so carboxyl, think acids. That's what we want to do. Yeah. Okay. So then amine or amino, amino has an N, has nitrogen in it. It's the only one with nitrogen in it, NH2. So we're looking for amino. Sorry. So we're thinking of building amino acids. If you can think back to biology and that circle structure where you had to be like UAG, and then it went to a specific amino acid, like cysteine or cystine. Oh shoot, can't think of it. I can't even say it. But anything glycine that ends in INE. I want to say the one and I just can't say it. Cysteine, I think something like that. But ending in INE is always showing us amino acids, and then that's going to be an example of the amine group for sure. Let's see what else. Okay, so then we have sulfhydryl. This only one with sulfur in it. It's sulfur, hydrogen, SH, sulfhydryl. And then this, we're looking at this big one here, is that we have these things called disulfide bridges. Not that you need to write that in your notes now, but you'll hear about them later. And it is using the sulfhydryl group in order to hold the structure of a protein. But we'll get to that in next chapter, but that is one of the main jobs of a sulfhydryl group. And then, oh yeah, these ones end in thiols. So we see ethanothiol, methanothiol, propanothiol. Any of those end in THIOL, that is going to be in the sulfhydryl group for sure. Okay, almost there. Okay, and then we have phosphates. Phosphates, a phosphate with 4 oxygens around them. When we're looking at this, I don't know, we have these this last one where we're underlining, we're looking at ATP. So ATP triphosphate, that has 3 phosphate groups on the end, 3 of those, cut off 1, then we get energy. So we have ADP, diphosphate, 2 phosphates. But then we can really think of nucleotides, which are the building blocks of DNA, which have a nitrogen base, you know, your A's, T's, A, T, C's, and G's. And then it has a sugar and phosphate backbone of your DNA. And that's another example of something that always, a nucleotide always has a phosphate, and that's going to attach to the next sugar down the backbone of that DNA. So these are making larger structures, and those are really the only things we can think of: ATP, ADP, nucleotides, that kind of stuff. I mean, we'll talk about kinases. I didn't even tell the class about it. Just focus on those ones. We'll talk about kinases later, which are phosphates, but we'll get there. Okay, then the last one that we're going to talk about is methyl group. Methyl group, carbon with 3 hydrogens around it. When we're talking about methyl groups, so one thing is that underlying DNAs, it affects the expression of genes that are in your DNA. And then the second thing was what I already talked about with those hormones, which we saw with the male and female hormones, and how it had the male hormone had that extra methyl group. So they're usually on larger structures, and then it's an addition or subtraction of those that's going to change the structure and then therefore change the function. So if we're going to remember something, think of male and female hormones and gene expression, and then it can change gene expression in your DNA for sure. And then, quickly, why do we study these? Because it's all going to go to our next chapter that we need to know. So go through those note cards. We did a couple examples that are on your worksheet as a class, and we circled some different functional groups within a large carbon structure, and then we named them. That is something that you're definitely going to want to know how to do, but like I said, that's in your worksheet that I gave you before you left, so you can practice those. And ask me any questions you have on Monday. But I kind of flew through that stuff. Hopefully you can stop, rewind. I don't know. Doesn't matter. You guys have a great weekend. See ya. Bye.